The overall goal of this procedure is to separate bacterial endospores from complex microbial community samples, such as sediment.
This method helps answer key questions in the field of microbiology on the presence and abundance of endospores in natural samples. It is based on a difference in resistance between endospores and vegetative cells towards chemicals and heat. The main advantage of this technique is that the method is non-destructive for endospores, so they can be used for downstream experiments like cultivation, quantification, and metabolic testing.
Although we have designed this method to work with the ecology of endospore-forming bacteria, it can also be used in fields such as exobiology or the food industry, in which spores can be either beneficial or detrimental.
After preparing solutions and equipment according to the text protocol, under a UV sterilized biosafety cabinet, use ethanol-flamed metal scoops to add 3 grams of sediment sample to pre-weighed sterile 50 milliliter tubes. Using a sterile graduated burette, add 15 milliliters of a sterile filtered 1% sodium hexametaphosphate, or SHMP, solution to the sample. With a liquid disperser or homogenizer and a sterile dispersion rotor, homogenize the sample at 17,500 RPM for 1 minute. Let the sample rest for 2 minutes and repeat the homogenization. Allow the sample to rest for 10 minutes for the heaviest particles to settle. Transfer the supernatant containing the cell